Right. Hi, everyone. My name is Sarah Cooperwich. I'm currently a PhD candidate working with Bala Chaudhry at Dartmouth College. Today, I have with us both Dr. Nancy Collins-Johnson and Dr. César Marín. We're going to be chatting on our What We Are Reading YouTube channel that gets shared with both the South American Mycorrhizal Research Network and then the International Mycorrhizal Society. So thank you both for being here. It's great to see you guys again. And yeah, it's great that we have both of the authors from the new paper of functional team selection as a framework for local adaptation in plants and their below ground microbiomes that was very recently published in the ISME journal.
Thank you for having us. It's really nice talking with you.
Yeah, it's an honor.
Cool.
Thank you.
Cool. So I guess just to start us off, what was kind of like the inspiration behind writing this paper? It's very clear that like it came from many, many years of like your own research and thinking. So I guess what made you guys sit down and work together and write this paper, and why now?
Go ahead.
I should start?
Yeah.
Well, I'm glad you noticed that it's from many, many years, because that really, that's true. And I think back and uh, back in the late 80s, I started to notice that the regular categories for mycorrhizal function as a mutualism just didn't work for the evidence that I was seeing, and a lot of other people were not seeing evidence for mutualisms, and it was clearly a lot more complicated than that. So over the last 40 years, I guess I've just been trying to understand what are the factors that control the outcome of this symbiosis. And it's not always a mutualism, but there are many important situations where it is a very important mutualism. In other situations, it's just the opposite. It can cause a growth depression. And so we really have to understand the mechanism of why it sometimes can be one or the other. So really, for the last, I guess, over 35 years, I've been trying to understand what those mechanisms are. And Cesar and I met at the ICOM meeting in Mexico in 2019.
2019, yes.
Yeah, 2019. And after my talk, he said that this is exactly what he's been very interested in studying.
Yeah, in my case, I was always very interested in evolutionary biology. I remember distinctively reading some books like sociobiology in 2009. So when I was in the second year of my bachelor, immediately I got interested into evolutionary biology, read a lot about it, did my bachelor thesis in evolutionary agroecology, was kind of the framework of the thesis. But then during my PhD, kind of abandoned evolution, did a mycorrhizal ecology thesis. So really with no evolutionary framework, but it was always in the back of my mind, like, and specifically the topic of multi-level selection. So when I saw Nancy's presentation in Merida, which invoked multi-level selection, I said, oh, finally, like from this 400 or 600 people, there is one person putting both things together. Because yeah, from my side, I started my first multi-level selection article is from 2015. So I have been writing about it for different types of public and audience, but never have put both things together. And I think this paper is the one that puts multi-level selection and mycorrhizal ecology really nicely in a single piece. Yeah.
Yeah, I definitely agree. I feel like reading it, it was kind of just turning like introductory biology like on its head of like things aren't just little unitary, units that are solitary and they're alive and they die. It's much more complex than that. So yeah, I really appreciated this paper putting both of those together for mycorrhizas.
Thank
So
you.
I guess like yeah, more recently the concept of like a holobiont has been kind of maybe more in popular science and in the news, especially with like human microbiome research. So I'm wondering how functional teams and functional team selection kind of differs from this more not traditional, but more popular, I guess, like holobiont term.
Yeah, well, you are correct that it's now like 20 years since 2007, 2006, since the holobiont concept was first introduced. Of course, there are some historical precedents, really important precedents like Lynn Margulies' work. But really, like, it became mainstream 2006. And since then, there have been several concepts of the holobiont that come from different disciplines. So there are concepts that come from the philosophy of biology, from quantitative genetics, from population genetics. Both types of genetics are different. I think our concept differs, and but although it captures some elements of previous concepts, it differs in the sense that we define a plant holobiont, but in reality any holobiont, as a complex adaptive system. So here in the figure, we can see what we mean by a complex adaptive system. Yes, thank you.
Can you see that, Sarah?
Yeah, it looks great.
So complexity science has been around for a long time, a century. And actually, some of the first complexity science papers come from biologists, from Sewell Bragg specifically. Uh, and as you said before, like turning around uh, the base biology that we all learn at college, I think this is one of the ways of really introducing complexity science and complex adaptive systems, well, in our case, to mycorrhizal ecology and soil ecology, but in honesty, it should be introduced to all biological research.
It should.
And one of the ways is to define holobiont as a complex adaptive system. You see the attributes there. They consist of a large number of agents that interact simultaneously through sending and receiving signals. They respond to signals through conditional action. Subgroups of agents create novel building blocks. Agents evolve over time and generate adaptations that improve. They are open and non-equilibrium systems, and they generate emergent properties in which the whole is different than the sum of the parts. If you read all those characteristics that have been used for societies, that have been used for termites or ants colonies, they definitely apply for a plant and its interacting microbiome. And I think that that's a big conceptual improvement in the definition of holobiont. And if you see in the figure, we include, although we are both mycorrhizal ecologists, but we include everything. We include eukaryotes, prokaryotes, and even viruses, because there is a lot of horizontal genes transferred through viruses. There is a lot that we don't know about viruses. So I think that they do play a big role in this.
Thank you. That was a good answer.
Yeah.
Yeah, that was great. Yeah, thank you. So I guess, well, I haven't even really necessarily given you guys a chance to like explain what functional teams and functional team selection is. So maybe we could take the next few minutes just to get, you explain it very clearly in the paper, but I guess just to get the most up to date and easily shareable to the audience.
4?
This one?
No, then the last one. Yeah, this one. 3, sorry. Yeah, figure 3.
This one. With
the decision, this one.
this tree.
You want to go ahead or?
Well, yeah, I can. And again, I just want to reiterate what César said, that our system that we were able to, gather these insights is a mycorrhizal plant system that includes all the other assorted microorganisms that are interacting within the holobiont of the plant. So there's always going to be a host that has there's some selection happening on whatever the host is that can be ameliorated by a team of microorganisms that is acting as a complex adaptive system. So the host can be a termite, it can be a human, it can be a plant. But in our case, we've used plants because we have such really good empirical evidence supporting this. But any host, if there is some sort of selection pressure that it could influence the host's success in survival and reproduction that can be ameliorated by microorganisms, that sets up a potential for a functional team. But in the process of writing this paper, we were forced by some very astute reviewers to become more clear on really what is the mechanism. And so we have proposed this hypothesized mechanism. And I think that we're going to be testing these criteria experimentally for many years to come. But this mechanism seems to work pretty well. So you can get a functional team if you have these 4 criteria. The most important is there has to be a selection pressure. And as I want to reiterate, it has to be some sort of selection pressure that has the potential to be ameliorated by a team of microorganisms working with it. The second is the host itself has to have the ability, a heritable ability to control its microbiome, at least to recognize the players and control the composition of the microbiome through either incentives, in the case of plants, like root exudates, or deterrents, again, in the case of a plant, where roots will just die and turn over and shut off. There's also chemical incentives and deterrents, but those are just examples. So the host has to be able to recognize and manipulate its microbiome. The third criteria is that within the surroundings of the host, there has to be enough microbial diversity to provide opportunities for functional teams to assemble. And this isn't probably a problem in most soil systems because they're astronomically diverse, and there's all kinds of organisms just waiting out in the wings. I've heard them called sleeping beauties. They're just waiting to be awakened into action with the opportunity. There's a lot of diversity just waiting in the wings, especially with bacteria and viruses. And then the last really important factor is there has to be enough time for there to be a sorting out of the teams so that a beneficial, functioning, complex adaptive system that increases the fitness of its host will actually build up in the surroundings. And for soils, this is really easy because soils don't move around. Plants don't move around very much in the soil. So you can get a buildup of populations of ecotypes, very specific ecotypes of bacteria, viruses, nematodes, all these little guys that can be building up their populations. And so that has to have enough time. If all 4 of those criteria line up, then we can expect to see functional teams and positive plant soil feedback, which is a self-reinforcing community of microorganisms that help the success of the host. If any one of these doesn't happen, then either no functional team will form or a negative negative feedback can form. And we see this a lot with plant growth depressions that are built up by microbiomes that are not selected to function together as a complex system, because all those organisms in that complex system, they all benefit when the host benefits because they have some sort of activity that they're doing together. So that's the mechanism we're talking about.
And I should add that, reiterate the previous point regarding that we here include everything, every microbe, every proteus, every archaea in the soil, particularly because of the empirical evidence. Well, a lot of the evidence, of course, comes from Nancy's work over the years, but there is also evidence from other laboratories around the world in which previously, in the previous decades, people isolated the AMF, the arbuscular mycorrhizal spores, and they found local adaptation. So the sympatric combination worked better. But it was clear that something was missing. And what was missing was the whole soil community. So then the experiments, very early on, like in the 90s maybe, the experiments were with all the soil microbiome, and it's still the same pattern of local adaptation was found. And as I said before, there are some very recent nice experiments in Rhizosphere. I saw one publication showing the exact same thing. So this, because of course in nature, in reality, it's not like the plant is interacting solely with the mycorrhizal fungi. It's
Oh, yeah.
and the mycorrhizal fungi by itself is interacting with a lot of other actors. So it's really necessary, this whole system view.
Yeah, I think it's especially exciting seeing these recent papers coming out about the importance of the hyphosphere bacteria. Hyphosphere means the bacteria
Mm.
and other actually archaea viruses in the surrounding the soil surrounding the hyphae. So there, a lot of very creative experiments now that are showing very important roles that they play in making a mycorrhizal fungus either beneficial or not.
And I think, coming to your first question, this discovery of the ionosphere was kind of what we needed. Like,
Yeah,
was an ignition point to really develop this. Yeah.
right. That's right.
Yeah. Yeah, that was some of the most, I think, like interesting and recent kind of research that was being presented last summer at the ICOM meeting. That was completely incredible because yeah, we're often taught that a mycorrhiza is just plant fungus. So it's great to see it getting all kind of shaken up like this. Okay, so for my next thought, this is actually a great figure to keep this on.
Okay.
This is more of maybe like an application or management type question, but I'm wondering like, are there situations in which negative plant soil feedback might actually be preferred? Like I'm aware in some like restoration type settings, you might actually want negative plant soil feedback to kind of encourage plant coexistence. So I'm guessing like, yeah, how might that work in kind of an applied setting? Like and then what role would a functional team play in negative plant soil feedback?
Oh, I completely agree with your suggestion in a restoration setting, because again, if you don't have enough time, and there's some excellent experimental evidence for this coming out of the Netherlands, where they showed that positive plant soil feedback is more common in late successional vegetation, and negative plant soil feedback is more common in early successional uh, communities. And plant communities. And so if your goal is to build up a different, site sort of community, you'd want negative plant soil feedback to move things along from maybe vegetation that might be more ruderal. And even we're seeing that, there's evidence for even the microorganisms tend to be more are selected early after, early in selection or succession than later in succession. So a lot of times in restoration, you kind of are looking to have some negative plant soil feedback at the beginning, and then over some period of time, get a stable community with positive plant soil feedback developing. Another idea where it might be useful is if you have an invasive species that's really taking over time, maybe you're kind of hoping that some more pathogenic relationships might take the invasive species out over time, and it somehow a lot of invasive plants are able to invade because they might commandeer what was more beneficial for the target native community. So I would say in invasive species management and in ecological restoration early on, negative plant soil feedback would be preferable. But in the situation of growing food, we'd probably and harnessing soil microbes to reduce our reliance on fertilizer, we would definitely hope for more positive feedback.
Great, thanks. Okay, so I'm wondering last, what direction you both hope this type of research will go in, and where you each see your own research in this field going.
Well, yeah, I think that there are many directions, and we are trying to encourage different people at different career stages to really have a look on into functional team selection, and particularly from different expertise. So molecular people, but also mathematical modelers, and of course empiricists. We have, I think, both empiricists and that's we should not forget about that. I think conceptually, one way of seeing the future is to determine, try to determine, although it's very difficult, kind of the boundaries of what a functional team is,
Yeah.
both in space and time, because the time scales of the plants and the microbes they are really different. So, for example, some bacteria can have 3,000 generations in 1 year, and 1 year can be nothing for some plant species. So how we handle that? 3,000 generations in other systems like in yeast are enough for speciation, for generating new species. So it's really difficult, and it still has not, in my view, still has not been solved how to handle this mismatch between the generational time of these interacting organisms. So I think that's one future step. The other step I think is definitely to play with these four requirements, or these four criteria, to modify them in experimental settings, and complement that with observations in situ, where we have natural variation of these things, of force, selective force and host constitution, microbial diversity, and time. So I would think those are some of the future plans. Try to establish a working group on this, and definitely the time has come for a joint effort that gets together people working on plant soil feedbacks, which is a lot of people, and really good research on that for 30 years now. But also people that has a more evolutionary expertise, and also people that works in the holobiome. So it's time to put them together. I don't know.
Yeah.
What are your future?
I think you summarized it very, very well. And again, this question of spatial and temporal scale is exciting and a little bit daunting as well, because again, these functional teams, they're non-equilibrium. And so as such, we don't really have a beginning or an end. And so I think that it could actually just be kind of describing how life just in general evolves not in isolation, and our ecological terminology of, you know, this is a mutualism, this is a commensalism, this is a parasitism, and then you see, you know, listed this fungus is a saprotroph now, and it's now this one is a parasite, this one's a mycorrhiza or a mutualist, and maybe that's a little bit too static. And we really need to put the organisms in their biotic context, and then put the biotic context in the abiotic context, and recognize that the whole system, the whole complex system, is moving together in space and time at multiple scales. And all that we see as humans are the emergent properties. And in our filter, our human filter, we see this as sort of static endpoints, but that's actually not reality. It's just coming through our human filter.
An analogy of that is that ever since Linnaeus, we biologists are really good at think in categories. But some people argue that, of course, as we know that how to delimit a species, but species evolve and lineages evolve. So even species are not static. The same in these ecological categories is I think is the next new challenge on how to not think in categories.
Yeah. That's just a human artifact.
Yes.
And one of the papers that really motivated me to, you know, in thinking about the functional team selection was actually a paper that was written by some astrophysicists on evolution at the system scale. And they coined, and it's cited in the paper, this group from Carnegie in Washington, D.C. They proposed this law of increasing functional information to describe the evolution of certain planets and the characteristics of planets and stars, that at a universal level, there's this law where, you know, things become more complicated with more information. And to me, that actually set up sort of the theoretical almost framework for functional team selection, because it is a constantly evolving, getting more complicated. It's just expanding with information. And of course, we think of information, you know, as genetic information and different combinations of genetic information. But then the complexity comes when you start having, you know, antagonistic and beneficial relationships emerging, and then what causes the stability of these relationships is really fascinating. So yeah, I'm planning to I'm so glad to have met César, because I was getting tired. Oh, woman, and I was getting tired. And so now César's really helped me focus what I'll be doing. I'll be retiring from teaching after in 2026, but I won't be retiring
Congrats.
from research. I'll continue to do research, and it will give us more time. We've been talking about pulling together a working group of diverse people with diverse backgrounds and interests, and we'd like to build up this working group. So
Yeah.
if I'm retired from teaching, I'll have a lot more time to do that. So that's my future plan.
Yeah, probably when we post this video, we will know more about this working group, definitely.
Yeah, that's great. That's super exciting. And that's it's nice to hear personally that there's room for less strict categories in biology. Because my background actually isn't in pure biology or biology really, so that was kind of a shock to my system almost, that I had to categorize things into like permanent categories and then like trick myself into believing they're real. So I'm excited that maybe I don't have to keep doing that for too much longer.
No,
Yeah.
it's a process.
Yeah.
And
Yeah.
the categories are an artifact.
Yeah.
Great. Great. Oh, that's great. This has been a really great conversation. Before we end it, does either of you have anything else to add?
I just want to thank you for taking the time to talk with us about this. It's been a real pleasure.
Yeah, thank you so
Oh,
much, Sarah. This was
yeah.
so
Thank you
interesting.
both so much. Yeah,
Yeah, thank you.
great.
That was it. All right. Bye-bye.
Bye.
Great. Thanks.
Stop.